Okay, welcome back, everybody. We are going to finish up the lymphatic system and the immune system today. Let me go ahead and share my screen with you. And when we left off on Friday, we were talking about the innate defenses. So these are non-specific defense mechanisms. These are things that we're born with, and they're ways of just preventing different pathogens from infecting you, right? So they include things like barriers that we talked about, and they include some internal non-specific defenses like macrophages and inflammation. In the next few slides, it goes over these innate defenses in a little more detail. The first of which obviously are physical barriers, trying to keep things from getting into the body. Also includes phagocytes, which are types of white blood cells that will chew up and engulf anything that is um, either harmful looking, or in some cases it is dead, or pieces of cells, dead cells, that kind of thing. Those include large phagocytes called macrophages, which are often found in various tissues. And the macrophages are there just to prevent different pathogens from affecting your body. Okay. We also have natural killer cells. Natural killer cells are a type of cells. Again, they are non-specific. They can recognize, generally, they're recognizing abnormal cells. So cells that are infected with something else, cells that are dying. And what happens is the natural killer cell can actually attach itself to the abnormal cell. So the abnormal cell has markers on the outside called antigens. Every cell has antigens on the outside, and the natural killer cell recognizes those antigens as something incorrect, and it attaches to or adheres to the abnormal cell. And when it does, the Golgi apparatus of the natural killer cell can release these molecules called perforins. What they do is they perforate the abnormal cell. They poke little holes in it. And by poking holes in it, it actually causes water to rush in and destroy that cell. Okay, it's still very much non-specific. These are just looking for abnormal cells. They're not looking for any specific pathogen or any specific cell. Interferons are part of our innate defenses. And interferons are defense against viruses. They're antiviral proteins. They don't actually do anything to the virus, but what they do is they block the virus from being able to replicate in the cell. Interferons are just a type of cytokine, so you've probably heard of them a little bit. There are different types. There's alpha, beta, and gamma. Interferon alpha is produced by cells infected with viruses. So they kind of put out the signal to stimulate natural killer cells to come to their aid and actually kill off that cell so that it doesn't replicate more virus. Interferon beta is secreted by fibroblasts, and it just slows down inflammation. And interferon gamma is secreted by T cells or natural killer cells. And again, it calls the other macrophages over to try to get those macrophages to chew up that cell that's replicating viruses. So they're basically just a way that virally infected cells can put out a signal calling other immune cells to the area to try to chew up that cell so it doesn't keep replicating the virus. There's a complement system. The complement system only works on bacteria cells. I mentioned this before. These are actually proteins that are found in your plasma and your bloodstream. And when they get the correct signals, they actually can combine together and form kind of like an arrow, right? And the arrow pokes a hole into the cell that has the antigen on it, whatever that pathogenic cell might be. And again, when you poke holes into a cell, water rushes in, and that can cause the pathogen to explode, to lyse. Other ways innate defenses work, we talked about inflammation, which is a localized tissue response to the injury, which causes redness, swelling, heat, and pain. Basically, the redness and the swelling, the heat and the pain, they're all by increased blood flow. They're all caused by increased blood flow to that area. And that increased blood flow is there to try to repair whatever the injury is, along with slowing the spread of the pathogen. You are mobilizing your body's own defenses against that pathogen. Okay? And we also have fever. I talked a bit about fever. Fever doesn't actually kill viruses or bacteria. It just raises the temperature and makes it less opportune for them, less optimal for them, let's say. So these pathogens would rather be at a particular temperature, right, at body temperature. And if you raise that temperature, they're not going to be as comfortable. In addition to that, it actually increases your body's own metabolism, which is going to increase the speed at which you can recover from these pathogens. So if a pathogen makes it past those innate defenses, and there's quite a number of them, and they do a pretty good job, they catch a lot of pathogens. If a pathogen makes it past the innate defenses, we have adaptive defenses. We have specific defenses. And these are no longer non-specific. They're no longer just going out looking for something that's wrong. They're actually looking for a particular pathogen, a particular virus, a particular bacteria. Okay, and these result from the coordinated activities of T cells and B cells. And guys, I'm sure have heard of T cells and B cells. Those are always involved in specific defense. So there are different types of T cells involved in this adaptive immunity. There are cytotoxic T cells, helper T cells, regulatory T cells, and memory T cells. If we just look at those words, cytotoxic, cyto means cell, toxic means kill or death, right? So cytotoxic cells can actually go out and kill, physically and chemically kill cells which are pathogenic. Helper T cells, right? The name helper tells you it's not really something that's going to go out and kill the pathogen. It will actually stimulate these cytotoxic T cells, and it will stimulate B cells, which we'll see what they do. Regulatory T cells moderate this whole response. We'll see how that happens. And you always have memory cells. So one of the hallmarks of adaptive defense is that some of the cells that you make up, right, when you see this pathogen, your body sees this pathogen, you clone these T cells and B cells, you clone memory cells, so that if they ever encounter that pathogen again, you get a much quicker, much stronger response. Always produces memory cells. Okay. Also involved in the adaptive response are B cells. Right? B cells pretty much are circulating through our bloodstream and our lymphatic system. They differentiate into what are called plasma cells, meaning they are in the plasma, in the bloodstream. They don't kill anything. They go out and they produce and secrete antibodies. We'll see how that works. Antibodies just kind of mark these cells. So they target these pathogenic cells and they mark them so that other white blood cells can come along and kill them and get them out of the way. And then we have natural killer cells, right? We talked about natural killer cells as part of the innate immune system. They are part of the innate immune system. You are born with natural killer cells. They are a type of lymphocyte. So they're listed here as well because they are the same type of cell, the same lineage of cells as T cells and B cells. Okay, and those natural killer cells can go around and actually, poke holes in pathogens and kill those cells as well. So here's a nice little flow chart showing T cells, B cells, and natural killer cells. They're all lymphocytes. Okay, lymphocytes are one type of white blood cell. They are the type that is involved in the adaptive immune response. And we have T cells, which you have these 4 different types of T cells. We have B cells, and we have natural killer cells. So the adaptive defense is kind of grouped into 2 different categories. There's these 2 big categories, there's 2 big buckets. There is the cell-mediated immunity and the antibody-mediated immunity. Cell-mediated immunity is provided by cytotoxic T cells. So cell-mediated, always T cells. Antibody-mediated immunity is always by B cells. So B cells always are producing antibodies. T cells are always going to be the ones that can actually go out and kill other cells and attack other cells. And so again, another flow chart, adaptive immunity. You can see you have this cell-mediated immunity where these T cells are activated, and the T cells can go out and directly and physically attack these cells. You also have part of adaptive immunity that's antibody-mediated immunity, which is where B cells can actually produce antibodies. We'll see that these Y-shaped proteins are the antibodies. We'll see what they do in a few minutes. Both of these lead to destruction of antigens, right? Destruction of these cells that have these antigens on their surface that shows the immune system that they are harmful, that they are pathogenic. One thing I want to point out here is that there's these arrows back and forth between cell-mediated and antibody-mediated immunity. If one is triggered, it can trigger the other. Okay, so if cell-mediated is triggered, it triggers antibody-mediated. Antibody-mediated is triggered, it triggers cell-mediated. They can trigger each other. They can work back and forth. It makes sense because you want the most coordinated attack you can have, right? You don't want just part of your immune system working. You want all phases of your immune system to go out looking for these pathogens. So adaptive immunity always involves antigens. Antigens are just little flags, little markers on the outside of the cell. That's it. That's all they are. So these T cells and B cells can recognize an antigen and recognize that it's something foreign. Your own cells have antigens on them as well. And these T cells and B cells can recognize the antigens on your own cells and leave them alone versus antigens from some other pathogenic cells. When these lymphocytes, T cells and B cells, recognize an antigen that is harmful, is potentially fatal, or not fatal, potentially harmful, they become activated. And when they're activated, there's a process called clonal selection that we'll talk about. They clone an entire army to go out and fight this pathogen. So you have all these different T cells and B cells kicking around in your body that are inactive, right? And each one can only recognize certain antigens. And we'll see how that happens in a minute. And when it does recognize an antigen, it goes ahead and becomes active and it clones itself so that we have an entire army of T cells and B cells that recognize this particular antigen. Okay. So again, we talked about innate immunity is present at birth. This is adaptive immunity. This is acquired after birth, right? Adaptive immunity can be categorized as active or passive. So active immunity develops when you are exposed to an antigen. So you get chickenpox, right? And the antigen or the virus that causes it is called varicella. It has a little antigen on its surface. Your T cells and B cells are exposed to it. They produce an appropriate response. Your body fights it. You get over it. You make memory cells. Part of what you clone are these memory cells. They stick around forever. And the next time they see varicella, the next time they see the antigen that's on varicella, they will recognize it right away. And you'll have such a strong and quick response that you won't even get sick from it, right? Because your body will respond before you even develop any symptoms. That's active immunity. Okay, and this even includes things like vaccines. In a vaccine, what they're doing is they are triggering your immune system. They're giving you a portion of a virus or a portion of whatever that pathogen is, and your T cells and B cells are cloning themselves to make a whole army so that if they ever see this particular pathogen again, they'll go out and fight that very quickly. That's different than passive immunity. Passive immunity is transferring antibodies from another source, not actually stimulating this person's immune system at all. You're just transferring antibodies to them, which will give them immunity for a short period of time. Then you have to transfer some more antibodies to them. This is done sometimes when people are, on their deathbed, right? Sometimes people are very ill with something like HIV or with cancer. This is also the type of immunity that a mother passes to her newborn infant when she breastfeeds. When she breastfeeds, her antibodies are actually getting transferred to the newborn. And so it's protecting the newborn because the newborn now has the mother's antibodies. It's not triggering the newborn's immune system to make its own antibodies, right? But it's protecting that newborn until that newborn child, until that infant starts to develop its own immune system. So again, forms of immunity, right? This goes all the way back to what we talked about last Friday. But part of our immunity is innate. We're born with it. It's non-specific. Part of it's adaptive. Adaptive is specific. It's something that we develop over time. And adaptive can be active or passive. Active happens naturally, right? When you are exposed to a particular antigen, when you get sick. And again, part of what happens when you get sick is you produce these memory cells so that you'll never have trouble fighting that particular pathogen again. Or it can happen artificially, right? This happens a lot of times. I think of vaccines as the most most common type of artificially acquired active immunity. The body is seeing some sort of a pathogen, usually a portion of the pathogen, but it's enough to trigger the T cells and B cells to clone themselves and make an army, so that if you ever see that pathogen again, you'll have this army ready to go. Okay. Another arm of adaptive immunity is passive. This is produced by transfer of antibodies from one source to another. This can be natural, such as when a mother breastfeeds her newborn child, right? This is passing her antibodies on to the newborn child. Or it can be artificial. If somebody, like I said, is very, very sick, they can actually be given antibodies. It can be administered antibodies from another person, just to protect them for a short period of time. So there are properties of adaptive immunity, right? There are things that adaptive immunity does that other types of immunity don't. One is that it's specific. All these other things we talked about are not specific. Each T or B cell responds only to a specific antigen and ignores all others. So going back to the example of chickenpox caused by varicella, you only have one T cell that's going to recognize that. All of your other T cells, however many millions there are, don't recognize it. That one that recognizes it goes ahead and clones itself. So now you have an entire army of T cells ready that recognize that particular antigen. And they also spread the word to B cells so that you have a whole army of B cells that recognize that one particular antigen. And that's it. They don't recognize any other antigen. They won't recognize the one that causes the flu or the measles or the mumps or anything else. Versatility. The body produces many types of lymphocytes, but each fights for a different type of antigen. So your body is producing literally millions of inactive lymphocytes, inactive T cells and B cells. They are specific to only one type of antigen. So each one of those T cells could only be activated by one antigen. So your body says, okay, these are all the things we might possibly come in contact with. Let's produce, all of these T cells and B cells. It's not until you actually come into contact with one of these antigens that your T cells will produce an army. Another property of the adaptive immune system is memory, right? It's always going to remember things. part of this cloning of themselves, part of the cloning of T cells and B cells, cloning this army and having it ready to go, is that you clone memory cells, which stick around for years. And the memory cells provide immunity against the new exposure. So if you're ever exposed to chickenpox a second time, and you've had it once and your body has fought it off once, the second time you get exposed to it, you won't even get any symptoms. Your body, your immune system will fight that off so quickly because it recognizes it very quickly and has an army ready to go that you won't even know that you had the chickenpox, right? You won't even recognize it because you wouldn't have had any symptoms. And then all adaptive immunes or adaptive defenses tolerate normal antigens. So I told you antigens are just little markers, little flags on the outside of cells, and pathogens have them, and your own cells have them. And a hallmark of the adaptive immune system is that your adaptive immune system can recognize your own cells versus pathogenic cells. So it recognizes these are your own cells, leave those alone, right? Not going to attack your own cells, it's only going to attack cells which are obviously not yours. So we'll start off with T cells. I said that you make millions of T cells, they're inactive until they see a particular antigen, right? So you've got all these inactive T cells, and when one T cell sees an antigen that matches, right? One T cell, it's going to clone itself so that you have an army of T cells. You have thousands or tens of thousands of these T cells, which have the same exact recognition of an antigen, of that same antigen. The thing is, T cells have to be presented with the antigen. In other words, if you have T cells floating around in your body, in your bloodstream, and you have a bacteria floating around in your bloodstream that has a flag on it, an antigen on it, the T cell won't bump into it and recognize it. What has to happen is a different immune system cell has to capture that bacteria and show the antigen to the T cell. It has to present it to the T cell. Okay, that's the only way the T cell will recognize it. This business on that says MHC proteins, those are your own antigens. MHC proteins are the antigens on your own cells. MHC stands for major histocompatibility complex. And all that means is that your own cells have antigens on the outside. They have flags on the outside. And these T cells recognize your own cells and they don't attack them. They leave them alone. So the T cells have to be presented with an antigen in order to clone themselves and fight off that antigen or that pathogen. There are two classes of MHC proteins that you should read about. They're found in different types of cells, but MHC proteins are just self-antigens. They're just antigens on your body's own cells. Okay. Antigen-presenting cells are other white blood cells. So here's what happens, right? One of the things that we talked about with the innate immune system is that you have phagocytosis going on. You have these macrophages going around like Pac-Man, chewing up things that don't belong. And this picture here is a drawing showing a cell that's just like that, right? This big cell, this big tan cell is a macrophage. This smaller oval over here on the left is a bacteria cell. And that bacteria cell has these little red circles on it. Those are the antigens, right? Those are the markers on the outside of the bacteria cell. So what this white blood cell can do, what this macrophage can do, it engulfs that bacteria cell and it chews it up. But it doesn't chew up the antigens, right? It doesn't chew up those little red circles. Instead, it takes the red circles and it puts it on the outside of its own cell. So now you've got this macrophage that chewed up a bacteria. And what it does is it says, hey, I just chewed this thing up. right? Here's what I saw. Here's the red circle that I saw. And it's showing that to the T cells. It's saying, look for this red circle. Anything with this red circle is bad news. It's dangerous. You need to go out and attack it. That is an antigen presenting cell. So antigen presenting cells are not some different cell type. It's just when a non-specific white blood cell can go out and do that, can go out and chew something up. And in chewing up that bacteria, it actually leaves the antigen alone, and it takes the antigen and puts it on the outside of itself. Once it does that, the T cell can recognize that antigen. So this inactive T cell, right, has a receptor on it. All T cells have these receptors on them. The receptors are really, really, really specific for only one antigen. And so that receptor that binds to, in this case, the red circle, is going to bind to the antigen, right? Only because that antigen has been presented to it by an antigen presenting cell. So when that antigen matches the receptor on the T cell, the two bind together, right? And the T cell binds that antigen, and then it actually clones itself. And in cloning itself, it makes an entire army to go out and to fight whatever bacteria have that particular antigen on their surface. Important markers, you've probably heard about CD8 or CD4 cells. CD8 markers are found on cytotoxic T cells. Those are the T cells that can go out and kill other cells. CD4 markers are found on helper T cells. Helper T cells activate CD8 T cells. They activate cytotoxic T cells. So what this is saying is that both helper T cells, which have CD4 markers, and cytotoxic T cells, which have CD8 markers, can recognize an antigen that's presented to them. They both have receptors on them that recognizes the antigen. If it is recognized by a CD8 marker, by a cytotoxic T cell, that T cell is going to go out and attack and kill whatever has that antigen on it. If it's recognized by a helper T cell, a cell with a CD4 marker, that helper T cell is then going to present that to a CD8 cell and activate the CD8 cell. There's also co-stimulation. Again, you want every aspect of your immune system to be firing, right? You don't want just T cells responding. You want B cells responding. You want non-specific cells responding. You want the best response you can get. So for the T cell to be activated, it must be co-stimulated. It has to be stimulated by, more than one thing, more than one antigen. So you can see that CD8 T cells, which are cytotoxic T cells, are activated by exposure to antigens on class 1 MHC proteins. And then what they do is once they are activated, they can destroy a target cell. So they can release perforins. Again, these are the molecules that perforate the target cell's membrane. They can go ahead and poke holes in that cell's membrane, which will allow like water to flow into the cell and it will burst open. They can also, once they're active, they release these cytokines. Remember, cytokines are basically chemical messages that go out and attract other white blood cells to come to the area, right? Sometimes they can activate genes in that target cell to trigger it to kill itself, which is apoptosis, is cell death that's killing, right? And sometimes they can actually secrete a toxin, which is poisonous to the cell. Bottom line is that these T cells can go out and kill other cells. These cytotoxic T cells go out and kill other cells. So here we have antigen recognition. You would have whatever this antigen presenting cell, this is a white blood cell that has chewed up either a virus or a bacteria and put the antigen from that thing that it's chewed up on the outside of itself, right? So it's presenting this antigen, this red circle. An inactive CD8 T cell recognizes that and binds to it. So now this CD8 cell is active. It goes from being inactive to active. It's an active T cell. It clones itself, right? So now you have an army of T cells with this specific receptor for this specific antigen. And in that army, you make memory cells, right? Which are inactive T cells, which will stick around for a long time. What happens is now this CD8 cell can go ahead and activate other CD8 cells, right, to go out and get the same antigen. You can see this one CD8 cell targets this infected cell. They dock, right? It links to the other cell with the infected cell with the antigen showing. And this CD8 cell, right, whatever this T cell is, can do a number of different things to attack that cell. That's what all these things are, right? These things are ways that it can attack that cell. So part of that army that it clones up are these memory cells, which stay in circulation. And so they will immediately become cytotoxic T cells if you ever see that same antigen again. So they secrete suppression factors, but they act after the initial immune response, and they limit the immune reaction to a single response. So that if you ever see this antigen more than one time, you're going to have a very strong response to it. If this antigen presenting cell activates a helper T cell, right? The helper T cells can secrete cytokines. So those cytokines will call over other immune cells. Helper T cells can activate cytotoxic T cells, and they will also produce memory cells. Right? So here you can see a helper T cell being activated by an antigen presenting cell. And then that helper T cell, when it clones itself, the clones start secreting cytokines. You also get clones that are memory cells, right? And you can actually activate other cytotoxic T cells. As for cytokines, there are these 6 major groups that you should know: the interleukins, the interferons, TNFs, tumor necrosis factors, phagocyte activating chemicals, colony stimulating factors, and miscellaneous cytokines. So I'm not going to go through those. You can read about what those are in the next few slides, right? Those are all just different types of cytokines, which are chemicals released by cells in the immune system. So when some of these cells get activated, they start releasing cytokines. And these different groups of cytokines do slightly different things. Bottom line is what they all do is they call over other immune cells, right, to help out with the destruction of this pathogen. So here's a summary of T cell activation, right? The antigen is bound to a class 1 MHC protein. This is an antigen presenting cell. This is where something like a macrophage has chewed up some sort of pathogen and taken the antigen from the outside of that pathogen, put it on the outside of itself, and showed it to a T cell. So it activates this T cell, which produces cytotoxic T cells, memory T cells, and regulatory T cells. So then what about B cells? Almost the same thing. As far as being activated, it's almost the same thing. But B cells are responsible for antibody-mediated immunity, so they can't go out and kill other cells. All they do is produce antibodies, right? That's how they attack their antigens. They are very specific. Again, they have each inactive B cell has a receptor on it that's really specific to just one antigen, just like the T cells, right? They have to be triggered by just binding to one particular antigen. Some of these antigens can be bound to class 2 MHC protein. So that's the same way that T cells can get triggered. In other words, triggering the T cell response also triggers the B cell response. You want both arms of this immune system to be activated. And so here you have, for example, an inactive B cell, right? It binds to whatever this antigen is, this red circle. Once it's bound, you get a sensitized B cell. And that's the same as an active cell, right? That's going to clone itself, and that's going to create an army of B cells. As I said, helper T cells can actually also activate or sensitize B cells. Sensitizing them is just activating them, just a different word. The activated B cell, so once it sees that antigen and it clones itself, you make a bunch of cells which become plasma cells. Plasma, because they're circulating through your bloodstream, right? They secrete antibodies, and the antibodies get into your interstitial fluid. They get out of the capillaries of your bloodstream and get into the tissues. You also produce memory B cells. So just like T cells, you produce these memory cells that kind of this inactive army that stays in reserve in case you ever encounter that antigen again. So what are antibodies? Antibodies are these soluble proteins. They're proteins that are dissolved in the plasma of the blood. And they're drawn as these kind of Y-shaped proteins. There are a pair of heavy chains and a pair of light chains on each antibody. There are some segments that are the same in all antibodies and some that are variable or different from one antibody to the next. So here you can see the Y-shaped antibody, right? This is the region where it would bind to the antigen. And what the antibodies do is they basically bind to all the antigens to keep the antigens from doing their job, right? To keep them from harming anything. And they cause the antigens to clump up, which is a process called agglutination. And we'll see that in a second. There are different types of immunoglobulins, right? Immunoglobulins are it's just another word for the antibodies. So there are different classes, these 5 different classes. IgGs are most of what we're going to be talking about. They are responsible for resistance against most viruses, bacteria, and toxins. IgEs are usually found in the allergic response, so like basophils and mast cells, which are part of your allergies. IgDs can play a role in the sensitization of B cells. So these can be something that actually activate other B cells. They're not the antibodies that go out and provide immune defense. IgMs are the first class of antibodies secreted after an antigen is encountered. So this is part of your first, your body's first site of this antigen. They'll produce IgM antibodies, and then they'll produce IgG antibodies every time thereafter. And IgA are a little bit different in that they are found mostly in glandular secretions. They attack pathogens before they gain access to internal tissues. And so IgA are found only in specific scenarios too. So the complex of an antigen and an antibody bound together, right, is just called an antigen-antibody complex. And here's what it would look like. So this brown cell here, this is some sort of a pathogen, something that can cause harm. And the antigens that are on the outside of it are these yellow circles. And in purple, these Y-shaped purple things are the antibodies. And so you can see that many of these antibodies many of these antibodies are bound to the different antigens. And what happens is now this cell, this pathogenic cell with all these antibodies bound to it, is circulating through your bloodstream and through your lymphatic system. And it's basically sending off signals to all these other white blood cells to attack it, right? These other white blood cells that attack things that don't look right, they're non-specific. They say, well, that doesn't look right. Having all those antibodies bound to that cell, that's not right. Something's not right about that. And so they will go ahead and attack that cell, try to gobble that cell up. I don't know why that keeps skipping ahead. Sorry about that. So a complete antigen, right? Exposure to a complete antigen, the whole entire antigen, will lead to the B cell sensitization and the immune response. And the antibodies will neutralize the antigen binding sites, right? You get a whole bunch of antigens binding to that one pathogen, or a whole bunch of antibodies binding to antigens on that one pathogen. And what it does is it activates the complement system. It attracts other non-specific white blood cells to the area, right? It stimulates inflammation. And so what it's doing is it's basically sending a signal out to all the other immune cells, all the ones that we talked about that are non-specific, telling them to come over and get rid of this cell to help out. And so here is, again, a diagram showing antibody structure and its function. What you want ideally is a complete antigen, because then you can get a nice complete antibody. But even if you get just partial antigen, you can still make antibodies against it and hopefully defend your immune system or defend your body. The first exposure is called the primary response. Every exposure after that is called the secondary response. So the primary response takes a bit of time, right? It takes a while for you to make these B cells, for you to clone these B cells, and then for the B cells to make the antibodies and for the antibodies to bind to the antigen. It takes a little bit of time. The secondary response happens a lot quicker. You already have an army of B cells ready to go, right? The memory cells are already there. And so you get this really strong response, and it's very quick, and you don't even usually get any of the symptoms. Right? So the primary response takes time to develop, right? The peak response can take a couple of weeks to happen. And you can see here the IgG antibodies and IgM antibodies. You can see that the peak takes about 2 weeks, right? For these levels to get to their highest point. Whereas the secondary response, you are going to activate memory cells. It's going to take much less antigen to trigger that B cell reaction, right? You won't need a lot of antigen molecules to trigger that reaction. And the amount of antibody produced will rise very quickly. You can see that it happens very quickly. Look at, how much quicker it happens. By 2 weeks, it's much higher. We don't really have any marks on the y-axis in this particular diagram, but the response and the amount of IgG that's produced is produced in much greater amounts, like 10-fold greater or 100-fold greater. And it's produced in only a matter of days instead of taking 2 weeks. So then we have to talk about immunocompetence a little bit. Right? Immunocompetence is one of those hallmarks of the adaptive immune system. It's the ability for the adaptive immune system to produce an immune response after exposure to an antigen. And there are all different ways that your body makes sure that your immune cells are immunocompetent. Right? They go through a series of tests, basically, before they get out into circulation and before they get out into the tissues, make sure that they recognize bacteria, to make sure that they recognize foreign antigens, that they can tell the difference between your body's self-antigens and foreign antigens. And so immunocompetence only means that they can tell the difference between your body's own cells and cells that could be harmful to you. Okay, and so here's an integrated summary of the immune response. I know there's a lot going on here, but in purple, this is your innate response, the non-specific response. Okay, all of these things are going to lead to destruction of antigen. But what I want to point out is that even like natural killer cells and macrophages, when they get activated, they can start activating the adaptive response, which is cell-mediated or antibody-mediated immunity. And guess what? Cell-mediated and antibody-mediated can activate each other. So even though they all lead to this destruction of antigens, you don't want to just have a pathway where it just only activates cell-mediated immunity. You also want to get antibodies involved. You also want to get macrophages involved. You want to maximize your chance at fighting this pathogen, whatever this pathogen is. So there's a lot of interconnectedness between these pathways. All right. A few other things to think about. Bacterial versus viral infections. Basic sequence of events in the immune response is pretty similar, but the initial steps differ. Cytotoxic T cells provide adaptive defense, where natural killer cells provide innate defense. Okay, and so again, if you have, here's some bacteria here, right? They are chewed up, phagocytosed by macrophages, and then presented to the T cells, right? By antigen presenting cell. That antigen presentation can activate cytotoxic T cells, which leads to destruction. It can also activate helper T cells, which activate B cells, which produce antibodies, which lead to destruction. And then if we think about viruses, what happens with viruses? Viruses infect tissue cells, right? If they infect the tissue cells, sometimes those cells that are infected can release interferons, right? Which causes like signals to other cells to come over and help the defense. Also, the cell that was infected will appear different, right? Its antigen will appear different. And so the antigen will appear in its plasma membrane, which will stimulate natural killer cells. And if this is an antigen presenting cell, can activate the T cells, right? And if it's infected an actual antigen presenting cell, like a macrophage, like a white blood cell, then it goes through again the antigen presentation, activating T cells, activating B cells, and still destroying the virus. So it's a little bit different. I mean, the end result's the same, but it's a little bit different. So when do we develop immunocompetence? Well, the fetus can produce an immune response at about 3 to 4 months. The fetal thymus cells migrate to tissues and become these antigen presenting cells. So they leave the thymus, they go to all these different tissues where they can chew up things that don't belong and show those antigens to the T cells. And in the fetus, the liver and the bone marrow produce B cells. But before birth, the maternal person, the mother, I don't know why I said it that way, the mother passes her antibodies through the placenta. And so the mother's antibodies provide immunity to the fetus. Now, this is passive immunity. Remember, we talked about active and passive immunity. This is providing immunity to the fetus, but it's not triggering the fetus's immune system. It's just the mother's immune system being passed to the fetus. And after birth, if a mother's breastfeeding, it's still passively providing immunity to the fetus for at least some length of time until, excuse me, until the fetus starts producing its own antibodies. And the normal resistance is that an infant will start producing its own antibodies through exposure to antigens, right? And the antibodies, the B cells, and the T cell levels will slowly rise to adult levels. There are different parts of immune competence, different things that affect immune competence, like stress and immune response. In stressful situations, your body produces glucocorticoids, and they do things like they actually depress inflammation. And so what that can do is that can limit your immune response. So in a stressful situation, your immune system is usually weaker. Now, glucocorticoids, those would be things like cortisol, right? You may have heard of people getting injured and having cortisone shots, cortisol shots. That's to suppress the inflammation. That suppresses the immune system as well. And then we get to, what about immune disorders, right? There's supposed to be this immunocompetence. Immune cells are supposed to be able to recognize self versus non-self. Well, there are times when that doesn't quite work. And that's where allergies, autoimmune disorders, and immunodeficiency diseases come in. Allergies are just hypersensitivities. So hyper means over, right? So these are just being oversensitive. Your immune system sees these antigens from a particular, whatever it is, and overreacts to it. So these are typical allergies, could be the pollen from different plants. A lot of us have seasonal allergies, spring and fall. It can be the coating of a peanut, right? One of the things in the coating of a peanut is now very allergic. A lot of people are very allergic to that. It can be bee venom. These are things which have allergens on this. They have antigens on the surface. Remember, every cell has a marker on the surface. And your body is recognizing that antigen as something that's potentially dangerous. It's triggering the allergic reactions. It's triggering inflammation. And it's triggering it at a really high level. And so that's what makes you stuffed up, right? That's what makes you feel like you're getting sick, is because your body thinks you're going to get sick from these allergens, or from these antigens rather. That's immediate hypersensitivity. That's type 1. That's what we commonly just think of as allergies, right? In some cases, this can be really, really dangerous, something like anaphylaxis, right? Some people go into anaphylactic shock, which produces swelling and hives on the skin. Your smooth muscles contract the respiratory system, which can make it really hard to breathe. But the biggest thing is that all of your blood vessels will dilate, which causes your circulatory system to collapse. It causes your blood pressure to drop really quickly. It's called anaphylactic shock. A lot of times when we feel allergies, or when we feel a little bit, a cold, a sniffle, we take an antihistamine. They're drugs that block histamines. That's why they're called antihistamines. So they can relieve a lot of the mild symptoms of allergies. So Benadryl is a good example of that. What happens is a certain type of white blood cells called mast cells release histamine, and that calls other white blood cells over to the area, and it causes your blood vessels to dilate, to open up and release more of these white blood cells. And so you need an antihistamine to kind of close your blood vessels or slow down the release. There are autoimmune disorders. So this is taking those allergens to another level, right? This is a situation where the immune system doesn't recognize normal antigens, or at least doesn't, or ignores normal antigens, right? It maybe even recognizes it, but it still puts out an immune response. What happens is B cells become activated and they start to make antibodies against your own body's cells. So there's, several examples of that. Type 1 diabetes is a big example. Rheumatoid arthritis is a really common example of that. Body's just fighting its own body systems. And then a step further than that are immunodeficiency diseases. There are diseases where your entire immune system is compromised and can't fight off very many things. One is caused by a viral infection from the virus HIV, which can result in AIDS. When the HIV levels get really high, it can result in something called AIDS. And what happens is AIDS severely suppresses your immune system. And so people who have AIDS and die usually die from a secondary infection. So they have AIDS and it destroys their immune system. What this virus, HIV, what it does is it targets the helper T cells. So we know helper T cells activate cytotoxic T cells and activate B cells. If you have like no helper T cells around, when you get something like pneumonia or something that most of us can fight off, your body can't fight that off anymore. Your immune system is just shot. It doesn't work well. People can become immunodeficient even from things like radiation treatments, right? From cancer treatments. One of the really severe side effects is that it's killing off a lot of your white blood cells. The effects of aging on the immune system, obviously the immune system diminishes with age. We become more vulnerable to infections as we age. Part of this is that the thymic hormone production is reduced. So thymic hormone is the hormone that causes T cells to recognize self versus non-self. T cells become less responsive to antigens. They don't necessarily always recognize an antigen that an antigen presenting cell is showing. And then there are fewer T cells becoming activated, which reduces the responsiveness of B cells. Remember, they talk to each other, T cells and B cells. And so then the immunosurveillance against tumor cells declines. You don't have as many cells that are actively out there looking for things that could go wrong. Right? And so how is the immune system integrated? Well, usually the nervous system and the endocrine systems are what can trigger the immune response or moderate the immune response. Right? So the nervous system are obviously nerves going from your brain to different areas of your body. The endocrine system is hormones. Those are glands secreting hormones, which can trigger the immune response. And so here's a diagram just showing how the immune response is triggered. So I'm going to leave that there for now. Okay, and I'm going to end on that note. If you guys have any questions, please don't hesitate to email me. Send me any questions you have. And I guess I will meet with you on Friday. But in the meantime, send me any questions that you have. Thanks. Have a great night.